This is a required practical from the topic B5, looking at the effect of plant hormones, in particular the hormone auxin, on plant growth. Now we'll just do a bit of introduction, remind ourselves about how auxin affects plant growth. If we remember that phrase, auxin in shoots speeds up growth, auxin in roots slows down growth, and we can link everything back to that. Okay, let's just look on the left hand side of the picture first at phototrophism. This is the response of the plant to light. So if the light was all around the plant, the auxin would just be evenly distributed in the tip of that shoot and growth would just go straight up. But in this situation, even at primary school, we learnt that plants go towards the light. We learnt that if we put a plant by a window, it will grow towards the window, grow towards the light. So the light is just coming from this right-hand side, and we can see that the tip of the shoot is growing towards that light. And what happens is when you've got that stimulus of the light, the auxin redistributes itself by diffusion, and it moves to the shaded side. You can see on the picture it says that the auxin is on the left side of the tip of the shoot. And what it does when it gets there, let's go back to that phrase, auxin in shoots speeds up growth. So it speeds up the growth of those cells on the shaded side. So if they're growing faster compared to the right hand side, it will bend the shoot towards the light. And what's really useful about that is that then those leaves will face the light, they'll be collecting more light, absorbing more light for photosynthesis. So we can see the build-up of auxin on the shaded side causes those cells to grow faster and the shoot bends towards the light. So let's go back to that phrase, auxin in shoots speeds up growth, makes those cells grow faster. Now we've got on the right hand side geotrophism or gravitrophism. The exam board go with either of those two words. I prefer gravitrophism, but it's up to you. Now here we've got two things going on, so a bit more difficult. They love talking about questions that are the plant is in the dark. So if the plant is in the dark, do not talk about phototrophism. Do not talk about where the light might be. Talk about where the light was. Okay, we're purely talking about gravitrophism when the plant is in the dark. Now this is a plant that maybe it's fallen over. You tend to see like palm trees, they're very kind of on their side and then they kind of go back up away from the ground, and that's because their root system is really weak and they rely on gravitrophism to kind of get them away from the ground and to take them back upwards. Now the shoot is growing away from gravity, so we call that a negative trophism, and the roots are growing towards gravity, so we call that a positive trophism because it's going towards the stimulus. Same thing in the shoot, okay? So the plant, for whatever reason, it's fallen over or it's in a hanging basket or it's been placed on its side, the auxin goes to the underside, to the lower side of the shoot. And it does the same thing it did in phototrophism, it makes those cells grow faster. So just here, these cells are growing faster, causing it to go against gravity back upwards. So that goes with our phrase, auxin in shoots speeds up growth. And then in the roots, the auxin also redistributes by diffusion to the underside or the lower side of the roots. Now, the phrase said that auxin in roots slows down growth. So rather than speeding up the growth here, like we did over here, this auxin here causes these cells to grow slower. That does not mean that the roots here are growing faster. They're growing at the same speed they were before, but now relative to the ones below, they are faster. Okay, so these are slower, these are at the same speed, but as a result, it would appear these are going faster by comparison. Hopefully that makes sense. So what would happen there is these roots would grow slower. Therefore, at the same time, these roots are growing down faster relative to this side. And so the roots would bed back into the ground. So auxin in shoots speeds up growth. The auxin distributes to this side, speeding up the growth. Auxin in roots slows down growth, it redistributes to this underside, slowing down the growth so that these roots bed back down into the ground. So that gives us our introduction to the concept of auxin. It's the hormone in plants that redistributes by diffusion to allow these changes in direction. Now we know we get those popular 6-mark questions for writing a method for any of the required practicals. I've put together a sample method for just changing light conditions, which is what we're going to look at on a computer model in a minute. Just looking at what happens if we've got bright light, reduced light, and then dark conditions. But as we know with all of these practicals, they can change the independent variable, mix it up, and we'll look at some variations after we've looked at this one. So I've got 10 seedlings. It's really important to do lots of repeats with this. If we just stuck to 3, we're probably not going to see a strong correlation in our pattern of results. We might get quite varied results, a bit like when we were doing the ruler drop, and you can get really quite different results, and then you look at your anomalies and you eliminate those. We're going to do the same process here. So we're going to have 10 seedlings in bright light conditions, 10 in reduced light conditions, and 10 in dark conditions. Now, at the beginning of this is like mustard cress, the cress that you have when you have an egg and cress sandwich. You buy it in a box, or you can buy seeds and grow them, okay? And they're not all going to begin at the same height. So in the computer model, you will see they begin at the same height, but in reality, that's not going to happen. So our method needs to... We're providing this method for somebody who has an understanding of science but has never looked at this particular investigation. So it needs to be detailed enough that they could follow it, and detailed enough for lots of people to follow it and be able to share their results. So they need a method that is informative enough that they can do it and they don't have to make assumptions for themselves. For that reason, we give actual values to the length of time we're going to leave it, the amount of water we're going to give it. So let's have a look. We measure the start height of each seedling using a ruler in millimetres. So we've got our resolution here. By saying we're doing it in millimetres, there's no hesitation. We're going to go to the nearest millimetre, not the nearest centimetre. That wouldn't be precise enough. So high resolution, measuring to the nearest millimetre, means that we've got precise results. We're going to leave all the seedlings for the same length of time. Now, rather than just saying that fixed length of time, we've actually got to give a suggestion. If we said something like 20 minutes, there's not going to be enough growth in that time to actually register a difference. So we're going to make a suggestion, and I've suggested 7 days. So don't be afraid and think, what would the exact answer be? So long as it is kind of in the rough region of being a suitable, realistic amount of time, that will be fine. So if you said maybe 2 days or a few days or whatever, I've gone for 7 days, okay? Water the seedlings, so that's one controlled variable. Water the seedlings with the same volume of water, and I've suggested 15 mils of water each day, okay? We could also keep them in the same temperature environment. That could be another controlled variable. You could suggest room temperature, or 20 degrees or 22 degrees C, whatever you wanted, but try and give a value to it. After 7 days, we're going to measure the end height of each seedling. So we're looking at start and end. Using that, we can calculate the increase in height by taking the end height minus the start height, and then we'll see how much it's actually grown in that 7 days. And then we can calculate the mean, but removing any anomalies. So we look at our set of results, and we'll look at our results once we've done the practical, and then we identify those that really don't fit the pattern. Maybe something went wrong, not a great seed, damaged in some way, didn't get, for whatever reason, didn't grow in the same way as the others, and we can remove those anomalies and then calculate a mean. Now, in exam questions, there's likely to be an anomaly because they like questions that relate to that. It doesn't mean that in every set of results we have to search out an anomaly. We can just say, actually, those results look like they will follow the pattern. We don't have to find one. Once we've calculated the mean, we can then compare our results and see how has light affected the growth of the seedlings. And we'll look at that once we've got our results. So I'm going to collect my results using a computer model, mainly because the practical is quite temperamental, it's quite difficult to carry out, so we're going to use the computer model so we fully understand the method. And this is a good model in the sense that we've got sunlight, we can change it to reduced light and then to the dark. and we can look at the growth of those seedlings over different days. And I'm not too bothered about the growth on, say, day 1 or 3 or 4. I'm looking at doing this over a fixed period of time, and I'm going for 7 days, as we saw in the method. So I've got 10 seedlings, and that's really useful because the more repeats I undertake, the more reliable my results are. And we can link this back to the quadrat sampling, where if we've got a big field and we're trying to sample that area, we're not just going to do 3 quadrats, we do at least 10, and it gives us a greater chance of getting a good representation of what that field looks like. Because if I only do 3 quadrats, I may well miss an entire plant species from the field. And in this situation, if we only had, say for example, these first 3 seedlings, they're quite varied in their heights, and it's not giving very reliable data. The other experiment that we do as a required practical, where we do multiple repeats, is the ruler drop. And that one, if you've tried to drop the ruler and catch it, it's quite temperamental. I think temperamental is a good word, in the sense that you get very different readings. If you only do it 3 times, you don't get particularly reliable results. And I've got myself here a results table, and I'm going to record all of my data on this results table, and the height of them in millimetres, and then we'll process that data at the end and conclude what it shows. So I've got a ruler here. It's going to take a little bit of time, but we'll be patient. We're going to go to the nearest millimetre, because I think otherwise it gets a bit confusing. I think we could go to the nearest ½ millimetre, but we certainly can't go any smaller than that. So we'll go to the nearest millimetre, looking at the height of these, and I'm going to add those to my table as I go. So number 1, 17. Number 2, 30. Number 3, 20. Number 4, 22. Number 5, 21. Number 6, 11. Number 7, 24. Number 8, 24. Number 9, 17. And number 10, 24. Okay, we'll have a look at calculating means in a bit, and we'll look at anomalies and what to do with our data to make it as valid as possible. Let's now take the days back down, and we'll go to reduce light, and then let's take the days up and see what happens after 7 days. Much taller now. Okay, we've got reduce light, so we've obviously got something happening here. There's a greater response to gravity. We'll talk about that later. So seedling 1, 37 millimetres. Seedling 2, 37. 3, 26. 4, 35. 5, 27. 6, 44. 7, 32. 8, 32. 9, 41. And 10, 40. Okay, so we've now done sunlight and reduced light. Let's take the days down. Let's go to dark. Let's take the days up. Okay, let's have a look. First test, first seedling there. 47. 2, 56. 3, 45. 4, 54, 5, 48, 6, 34, 7, 44, 8, 7, 8 is here, 52, 9, 33, and 10, 40, I don't know if that's 41 or 42, let me check, 41. Okay, so we have now 3 sets of results. Okay, so here are my results from the computer model that we just looked at. And I don't have to find anomalies. Like I said before, I don't have to search them out. But if I do have obvious anomalies, then I don't want to be including those when I calculate my mean. And I'm thinking, probably looking at the sunlight, we've got this quite short seedling at only 11 millimetres. I'm going to circle that to represent it as being an anomaly. And potentially, I think I could go for this number 2 at 30 millimetres because that's quite out of the range. Now let's have a look at calculating my mean. I'm not going to include my anomalies when I calculate my mean. So rather than adding up my results and dividing by 10, I'm going to divide them by 8 because I've got 8 results. So 17 plus 20 plus 22 plus 21 plus 24 plus 24 plus 17 plus 24. Must press equals, and then I'm going to divide that by 8. Now the result I get is 21.125. What's really important and a good way of getting those extra marks is that we keep the same number of decimal places. So there's no point trying to be fancy and adding in a greater number of decimal places. So I'm going to take my 21.125 and I'm going to round that to 21. So there is my mean. Let's do the same. So look through my results. Do I have any anomalies? Quite a big range, but I don't think I can call 26 an anomaly because I've got a 27 as well. I don't think I can call 44 and 41 or 40 anomalies because they're all quite similar together there. So whilst we've got a big range, I don't think there's any obvious anomalies. So 37, add 37, add 26, add 35, add 27, add 44, add 32, add 32, add 41, add 40 equals, and then divide that by 10 because I've got 10 repeats, and it's 35.1. So I'm going to call that 35. Last one then, let's have a look at the range. So we've got a 33 and a 34, and we go all the way up to 56, and we've got a 54. I'm going to leave those results as they are, not identify any anomalies. So 47 plus 56 plus 45 plus 54 plus 48 plus 34 plus 44 plus 52 plus 33 plus 41 equals, divide by 10 equals 45.4. So I'm just going to call that 45. So we just look at our mean data now. We can see in bright light the mean height of seedlings is 21 millimetres, reduced light 35 millimetres, and dark 45 millimetres. Now what people attempted to talk about when they analyse this data is that in the dark situation, that the plants are growing tall, they're trying to get to the light. They're not trying to get to light because there is no light. They're not going to where the light was or where they think the light is. It is all down to geotrophism or gravitrophism, depending on what you want to call it. So If you imagine when the plant hormone auxin can respond to light and gravity, it's responding to two stimuli there. It can also respond to water as well, but we won't talk about that one because it's not in the course. So when the auxin is responding to two stimuli, it's kind of splitting its distribution based on those two stimuli. You remove one of those stimuli, in this case the light, and the plant can only respond to gravity. So we know that gravitrophism is about the auxin redistributing in the shoots to make the shoots grow as quickly as possible away from the gravity. It's a negative response to gravity, negative trophism of the plants growing away. So they grow that much taller when it's dark because all of the auxin is purely responding to gravity, not responding to light. Hence they're much taller in the dark. Hopefully that makes sense. Now, I said earlier on we might have an alternative method. What we've looked at so far and what our method looked at, what our investigation looked at, was purely looking at light intensity. Now, that light intensity could be, alternatively, moving the light at different distances, just like we did in the photosynthesis practical, where you have the lamp at, say, 0 centimetres, then 20 centimetres, 40, 60, and so on. Alternatively, we can look at different options for this experiment. We can see this here, okay? So we know that the auxin is in the tip of the shoot. Plant A, normal plant, as we've already seen. There's the tips of the shoot are there, the auxin is there, and A is likely to bend towards the light because that's how it's going to respond. Just like we talked at the beginning, the auxin will go to the shaded side, make those cells grow faster, and it will bend towards the light. Now, B has had the shoot tips removed. That means that the auxin has been removed. Now, it's the auxin that allows the shoots to grow, so B will have no growth. Now, C would be really, really tricky to actually experiment in real life because the crest is tiny, okay? But if we could make little tin foil caps and put them on the top of the shoot where the auxin is, then the auxin won't be able to respond to the stimulus. It will effectively be in the dark, even though the rest of the shoot is in the light. So we would expect C to continue to grow straight up because the auxin cannot respond to the stimulus. Let's have a look at an exam question that I found. Similar sort of thing, one more variable added in there, and that is A. And A has put aluminium foil around the bottom of the shoot, so around the stem but not in the shoot tip. And we can see that that makes no difference at all. It still goes towards the light because the auxin is found in this tip of the shoot. So that one was unexpected and not one that we have seen up till now, but it does actually respond in the way that we would expect it to because we know the auxin's in the tip of the shoot. B, we've just seen with the aluminium foil on the tip there, so that's not allowing the auxin to respond to the light stimulus, so we can see it's grown taller just like in our experiment because it's just responding to gravity. And then with the tip removed, no growth, that's plant C. And then just as it is with plant D, you can see it's bent towards the light, which we would expect. I've got one more exam question, which is a bit of a different one, something that we wouldn't necessarily expect to see. Again, we must see that this plant was left in the dark, so it's not responding to light in any way. And we've got these shoots that are currently, they've been grown so that they're growing kind of off to the side here. We've got damp blotting paper. Blotting paper is like filter paper, okay, so it's damp, so there is water in every direction. Now, I said earlier that there was the response to water, which is hydrotrophism. So the reason that they've put water everywhere is so that the plant doesn't respond to water, okay, it's just responding to, in this case, gravity. Okay, so we've got in A, it's stationary, it's not moving at all, so we would expect the shoots in this to grow against the gravity, negative trophism, and start to curve up. Now B is being used as a control. Now, to try and eliminate the effect of gravity, they've got it on a motor slowly rotating. So at any one time, whilst the auxin could redistribute to the effect of gravity, because it's then moving again, it can't keep up, so it can't redistribute based on gravity. So this is effectively, I call it like an anti-gravity machine, okay? It's allowing the shoots to not respond. That's probably poor English, I should say. It's not allowing them to respond to gravity, okay? So it's meaning that they're just going to probably grow straight outwards as they are, because they're not responding to gravity in that way. And we've said it's in the dark because they can't respond to light, and we've got water everywhere, so they're not responding to the water. So the only thing they can do, is grow in that straight direction towards the right. I think I've looked at all of the different possibilities for this method. Be careful with what the aim of the experiment is. What is the independent variable? Write your methods so that it matches what your independent variable is. We've seen some results. We've looked at anomalies. We've talked about precision. We've talked about reliability. So let's just look at those words again quickly. Precision. We're measuring to the nearest millimetre, which is more precise than measuring to the nearest centimetre. We talked about that being high resolution. We're doing 10 repeats to improve the reliability of our results. Now, on some rulers, we see that the scale doesn't really begin at the end of the ruler. If we look at this ruler here, we can see that the scale actually begins some distance in on the ruler. Now, if we were just to measure that, the height of the shoot, then it wouldn't be accurate while we included this part of the ruler. So we need a ruler that starts like a meter stick, really, starts on the 0, and then we're going to get a more accurate reading of the height of the seedlings. Now, if we're doing the same way of measuring and we're doing start and end, we would eliminate it anyway. But if we're just looking at getting an accurate measurement of the height, then we've got to use the ruler in an accurate manner, okay? And having a ruler that starts with the edge of the material of the ruler on 0 is going to enable us to be more accurate in our readings. Hope that makes sense. Right, so that is everything that you need to know about the plant hormone practical in topic B5, paper 2. Hope you found it useful. Thank you.